what we now need to go on to is some of the more up-to-date parts of the inheritance part of this unit. And we're going to start with a bit on DNA technology and genetic modification, if you like, and then move on to a bit on gene probes, which I thought was going to be quite big, and then when I looked at it, actually it's not. It's actually quite a small bit, really. We'll then look, I think, at the control of gene expression, and then we go into genetics, and that starts to become a little bit more organized again. So this is it's a very flexible bit, and one of the reasons is that this actually provides quite a lot of potential material for the presentations. So I don't want to go into too much detail on anything, plus I haven't got the time to go into too much detail on anything, plus I'm not an expert enough on a lot of this stuff because it's very current, because it's technology, and it's not the sort of thing I've ever done, you know, in terms of my own studies, that I've not had hands-on experience. So all you're really getting is, you know, the theory stuff. Hopefully, some people will choose to give us a bit more detail in the presentations. So the first bit of this is, well, it's going to sort of dip in and out of various bits, really. Recombinant DNA technology. Recombinant I'm putting in brackets because it's not just about recombinant DNA. Recombinant means new combinations of, you know, where you take a genome in an organism and you might add another gene, or you might edit a gene, change a gene. And so recombinant DNA is DNA that's different from what you started with. It's been recombined. But DNA technology generally, you know, is broader than just that. So, for example, DNA probes that I was talking about, it's a technological part of this, but it's not about creating new genes. So this part of it leads us to quite a lot of potential material for the assignment, and I will give out the assignment this morning so that you've got something to you know, base your thoughts on. Now, I've had ideas from several people about what they want to do, and you know, if those are still your choice, then you know you have your name against those choices. But if you change your mind, that's fine. It doesn't matter, you know, as long as somebody else hasn't already got what you want to do, then the choice is up to you. Now, the sort of starting point with this then is, you know, what are we actually using this for? And you can approach this in all sorts of directions. You can either look at the examples first or look at the technology first. And I just want to introduce a few of the examples so that you can see why this has been developed. Now, if you go back to the first transgenic organisms, we're looking at, I think, the 1970s. Was actually the beginning of all this. So it is actually quite a long time ago. But of course it takes ages, it takes years and years and years to go from the first time somebody manages to do something to it actually becoming you know, a viable and usable technique for anything. So there are terms that we come across here which are just worth noting down. These are not really notes that will make a lot of sense. They're not going to run in any sort of particularly organized sequence. You do have in your handouts, following that practical instruction and the questions, 2 handouts which are those bio fact sheet ones. They are fairly elderly, actually. The first one, genetic engineering, from September 1997. So yeah, 20 years, that one. And modern techniques in biology, genetics, from April 2000. So again, 17 years, you know. So these are not up to date, but they are the basics of a lot of the techniques. So that's what we have to focus on. And then specific examples, I hope, will be the sort of thing that people choose to do in presentations. Consequently, I don't really have a plan for how I go about this. I just sort of do various bits and pieces and hope we've got enough by the end of it. So what we're looking at is, well, you can call them transgenic organisms. All right, that's one piece of terminology. Trans obviously means transferring or from one to another. You know, it means transgenic means having a gene from another organism. You also come across the term genetically modified organisms, which I think we use more frequently, don't we? Just as its abbreviation, GMOs. Yeah, or we talk about GM stuff. So you have genetically modified organisms, which I think means the same thing. There may be subtle differences, but genetically modified organisms. And this is what we tend to refer to, you know, when we're talking about food, perhaps. You know, that a lot of people have issues, don't they, with genetically modified food. So, how much GM food do you think we grow in this country? None of it. We don't, yeah, we don't have genetically modified foods in this country. If you have food, if you eat food that has ingredients from elsewhere in the world, you might. So particularly if you've got soya protein from America. They use genetically modified soya in America, and they don't separate GM from non-GM. So anything from America that's got soya in it, or soya from America, I should say, is likely to contain some GM soya. But in this country, we don't grow any genetically modified crops for human consumption. It's all trials and things like that. And because it's part of my husband's job, is to actually look at the ingredients that go into a lot of food products, and he has to look at what they call the traceability of ingredients, and, you know, confirm that there is no GM products in there. So yeah, we don't have any. And for a while, it was one of those things. I think the company Iceland were making you know, quite a feature of it in their advertising, that they didn't have any GM products. But, you know, genuinely, if it's UK based, it won't have GM in it. Is that a good thing? Well, it depends what you understand GM to be and what you understand it to be there for. There are a lot of uses for GM, most of which are intended to be good. You know, people don't just genetically modify for the fun of it, because it's not fun particularly. It's actually a very, very tricky process. It's expensive, and why would you do it? It's actually done for very, very distinct purposes. For example, you can increase the yield of a crop by genetically modifying it. By inserting a gene from, say, a different variety of the same plant, you could get a crop to produce twice as much grain, perhaps. So would that be a bad thing? Well, no, because obviously, you know, we've got increasing pressures on feeding the world, haven't we? So increasing yield is a good thing. You also have, and I put a couple of examples on the list over there, herbicide-resistant crops. Now, theoretically, I mean, there's good and there's bad with this, because the idea is, and this is what the soya will be from America, it will be what they call Roundup-ready soya. Roundup is you might have heard of it, it's a weed killer, yeah, a pretty extensive, sort of non-selective, sort of, it'll kill everything, pretty much, yeah? You can use it to clear areas of land. So what they do is to modify the crops so they're resistant to Roundup. In other words, Roundup doesn't kill the crops. So what does it kill? Everything else. So you only need to spray your crops once with Roundup, and you get rid of the weeds. That improves your yield, and you don't need to keep spraying with other crops. So you're using less sprays, you're using less herbicide on your crops. The problem you have, yeah, is that the genes, if you get your crops, your resistant crops, into breeding with wild versions, because there's a lot of wild versions of crop plants, then there is the risk of transferring that resistance into the weeds. So, yeah, you know, potentially an issue there. You've got things like pest-resistant crops. things like, for example, cotton. Cotton is a bit prone to being eaten by it's a grub. It's called the boll weevil, right? The boll weevil produces little grubs which burrow into the cotton pods and destroy the cotton fibres. So, you know, you can have complete cotton crops wiped out by this. So they've introduced a pesticide into the genome, or a gene for a pesticide, a natural pesticide, into the genome of cotton, so that the cotton now can actually repel these insects and yeah, get rid of them. And yeah, so you don't have to spray them with pesticides. So again, it's reducing the amount of spraying that you have to do. So, you know, there are things like that. You also have, and I've got on there, golden rice as an example. Now, I'm not sure that this has taken off, and it's a cost thing more than anything else, but there are large parts of the world where rice is a staple food, yeah? You've got, you know, if you go to the east, particularly sort of India, China, that part of the world, a lot of rice has been eaten. But you also get quite a lot of vitamin A deficiency. Now, vitamin A can be made in the body from a yellowy orange pigment called carotene. All right, hence, yeah, carrots help you see in the dark because, yeah, good source of vitamin A in the carotene, which is then used for your eyes. But there's quite a, you know, an issue in some parts of the world where they eat a lot of rice, that they have a vitamin A deficiency. So they've tried very successfully actually to put a gene, and I think they take it from daffodils. The gene for carotene is inserted into rice, into the genome of rice. The rice then produces carotene, hence it's golden rice. It's rice with vitamin A, it's fortified. But the problem is you've got the cost issue, because that can only be bought from the big companies that actually produce it in the first place. You're not, and I suspect it's sterile and probably unable to reproduce itself, so that you have to keep buying it from the company. So, you know, it's not going to work for your poor village farmers, is it? And all that sort of thing. So there's a huge amount of ethics associated with all this, obviously. So modifying organisms is intended to produce better yields, to produce pest resistance and herbicide resistance, so you can reduce the use of chemicals. And there was this, where did I read it? There was an article talking about the concept of organic GM, yeah? Which is an interesting idea, because immediately somebody says organic, you think, oh, this has not been tampered with in any way, no chemicals on it. But actually, in order to achieve that, the best way is to genetically modify. So there was this, I think it was just, you know, somebody raised the question, well, you know, can we be aiming for, organic GM, which is a, you know, it's a nice concept, that one. As far as the examples are concerned, though, I've given you all plant ones, haven't I? You know, rice and cotton and soya and all that. So do they genetically modify animals? Limited success. It's a bit harder in animals, but it is possible to genetically modify, and I can't think whether they use sheep or goats, and insert the gene for human growth hormone into sheep or goat, well, it'd be egg cells, I assume, next to the gene that produces milk protein, so that when the milk protein is expressed, you also get growth hormone. So these animals produce growth hormone in their milk, and then all you've got to do is extract the hormone from the milk. Because otherwise, where do you get growth hormone from to treat people who have issues with their pituitary gland? You know, it's dead bodies, basically. You'd be looking at pituitary glands and extracting it from there. So, you know, actually producing it in the milk of animals, you can see how useful that is. And that comes under that category, my second word on there, farming with a PH. Pharmaceutical farming, you see, producing animals to produce medical products. The other one that I've got on there, human insulin from GM bacteria, that's probably one of the best known examples. That even comes into GCSE biology, that what they do is to get the human insulin gene. Now this is going to lead me into where from, You get the human insulin gene, you cut open one of those little loops of DNA that you get in bacteria, the plasmids, put the human insulin gene in there, get bacteria to take that up, and then the bacteria will just churn out human insulin. And that is how human insulin is produced for people with type 1 diabetes. So there, we've only got, half a dozen examples at most. Now, there are dozens and dozens of them, if not more. And, if you're interested in doing a GM example for the assignment, then, you will, need to track down a bit about the technology of it. But there's obviously, it's a good topic for ethics as well. Theoretically, then, shouldn't you be able to genetically modify humans? Because, we're not all born with a perfect set of genes, are we? Some people are born with a genetic condition, which, well, couldn't you correct that? Yes, you could, theoretically. But you're not allowed to. By law, you can't genetically modify people yet. Right? It will probably happen, and it will use a particular technique that's a relatively recent discovery, which Pete's wife did her presentation on last year, and that's one called CRISPR. But you can't actually produce genetically modified people from embryos at the moment. You can try and genetically modify people who are already mature and have the say-so, but you couldn't genetically modify every one of the 30-odd trillion cells in the body. However, if you look at something like cystic fibrosis, who's doing CF? Yeah. Then you'll look at gene therapy, I suspect, in that, because the idea is that you can genetically modify the cells of the lungs, which are particularly affected, and just correct the genes in those, and one of your main issues is solved. But there have been other examples of genetically modified people in order to cure conditions. There's one genetic condition called severe combined immune deficiency, SCID, which essentially is like AIDS because it knocks out the immune system. And there have been cases, it's a sort of boy in a bubble type condition, where people, you have a child who just has to be kept inside a completely sterile environment for their lives. If you take some of their bone marrow and modify the genes in the bone marrow to produce, the normal immune response and put it back in, I believe that's been used as a treatment, and there will be others. There will be more and more as time goes on. So you then come to the issue of possibly designer babies. Can you tinker with, can you choose, can you change things about, your potential baby? At the moment, there are things that you can select for. If you have IVF, you can select male or female if it's done for family balancing sometimes, not so much in this country. You can go to America and that's not a problem. But if you've got several boys, you can choose a female embryo. As somebody told me yesterday, the Beckhams did it, I gather. Yeah, the story had got inflated somewhat, and they said, is it true that Victoria Beckham chose the eye colour and the hair colour? No. And when she looked it up, they had gone to America to choose to have a girl. But that is as far as it goes, because other genes, you can't even find them that easily and select. But you can also select, there's a list of conditions that you could select embryos on the basis of, and these would be conditions that would cause severe suffering and would be life-limiting, that sort of thing. And so people can choose, to have an embryo that doesn't have haemophilia, say. I believe you can select an embryo that doesn't have cystic fibrosis, and several other conditions as well. You can select an embryo that doesn't. But you can't change the genes of the embryo. Right? You can choose what you've already got, but you can't change them. So what we're looking at to start with is this idea of changing DNA. And then tomorrow we'll look at how you might actually identify where a gene is using a gene probe, but that actually is not terribly complicated. Right, so transgenic organisms, or GMOs then, produced by changing the genes of an organism, and it might be from one of the same species, which I think we feel more comfortable about, don't we, than changing it with a gene from another species. So let's say you're trying to modify a crop to produce a greater yield, then if you just perhaps inserted a gene from another of the same crop but a different variety, we wouldn't feel too bad about that. But if you've got a tomato plant and you want to have vine-ripened tomatoes, but you want to be able to keep them in cold storage, which at the moment you can't because they just go squishy, ripe tomatoes, don't they? You could insert the gene, the antifreeze gene of a deep sea fish. You might remember doing that when we did the function finders activity with the proteins. And people are not so keen on that. You know, a fish gene in a tomato plant is a little more disconcerting than, yeah, just keeping it within species. So, you know, this is the sort of thing that people don't really understand. And people get into these debates, and, you know, one of the most ridiculous debates, of course, hinges on the fact that, well, you know, we shouldn't be playing God, right? Now, you don't have to have any particular belief to actually see the flaw in that, because if you do, then you should see it that actually any knowledge and ability that humans have is God-given. So if you have the ability to modify genes, and you have the ability to do medical research, and you have the ability to invent nuclear power, not invent, you know, use nuclear, and you have the ability to do this and that, you should do it. So that would be my argument, would be that, you know, if a human has a God-given ability to do it, then it's not wrong. Yeah, there are certain things that you would, yeah, perhaps find a little hard to argue the benefits of. But if you are doing something which has, for its intended outcome, a benefit to humans, or any other organism, to be absolutely honest, then, you know, why shouldn't you do it? You know, it's not playing God, is it really? It's you know, it's doing something you have the ability to do, and you know, that shouldn't really be wrong, should it? However, you know, the ethical side of it is critical. The testing, the safety, all that sort of thing is critical, and nothing should be done without checking that it's safe. So, you know, you sort of have to separate the arguments out. But people will have very extreme views sometimes, or just in the kindest possible sense, ignorant views, you know, because they don't know, because they don't understand what the process actually is. One of the problems is that if you insert a gene from one organism into the other, you can't be sure exactly where in the genome it's going to go in, always. And you could disrupt the function of another gene, and that is one of the biggest issues, that you know, you can't just sort of stitch it on the end of a chromosome or put it in the middle and assume there's a nice space there for it. That, you know, it could cause other problems, and particularly now that we know that the non-coding DNA between our genes has an awful lot more functions than we realise. You know, if you've got say, you know, a sequence of I don't know, 1,000 base pairs that, you know, you don't think it's doing anything, and you stuff a gene into the middle of that, yeah, you might disrupt the expression of another gene somewhere else. So it's a tricky thing, you know. I'm not saying it's all truly wonderful and we should do as much of it as possible. It is something that, yeah, has got a lot of scope for development. Right, first thing that we actually need to do then, if we're going to insert a gene, we've got to make the gene. We've got to get a gene from somewhere, haven't we? So the first issue is obtaining the wanted gene. So it might be, for example, the human insulin gene. So whatever gene it is that we require, how are we going to get it? Well, there are several ways you can do this. A gene, if you remember, is just a code for making a protein. Now, the protein, we know the sequence of amino acids, so we could reverse engineer that, couldn't we, and say, well, in order to make this protein, then we need this sequence of bases. So you can actually just put together a piece of DNA in the lab. It's not that hard to actually, reverse engineer the gene that you want. Well, you can find the gene and say, well, it's this sequence of bases. It's adenine, adenine, thymine, cytosine, cytosine, guanine, cytosine, guanine, cytosine, guanine. Well, just put that sequence together in the lab and there you are. We do have equipment for doing that. So the first option is simply create in the lab. And that's an option. I give the impression it's a very easy thing to do. It's probably not quite as straightforward as I've suggested. The second option, and these don't come in any particular order, would be to cut it out of a DNA sample that we already have and we know where it is. So the second thing would be to cut it out from the donor organism's DNA, really. And you have to do this using something rather special that I would like to spend a bit more time on, but we'll see how things go. There's a particular sort of enzyme. And these enzymes are called restriction endonucleases. Now, there's loads and loads of these, and what they do, the restriction bit means that they cut in a restricted way. They cut restricted lengths of DNA. Endonuclease, endo means inside, doesn't it? And nuclease means nucleic acid. So these cut inside nucleic acids in very restricted areas. Now, we know how stereospecific enzymes are. And these cut at very specific base sequences. All right, so I'm going to end up, this is a long sentence, but these cut at specific base sequences. And if I can come back to that at some point, I will. These enzymes, these restriction endonucleases, have been extracted from bacteria. Why might bacteria have enzymes that cut up DNA? Well, it's part of a bacterial defense system. What might a bacterium need to defend itself against? Sorry? No, not the immune system and T cells. If it's going to cut up DNA, what might get into a bacterium? It's interesting, nobody gets this. Can bacteria get infected? There's viruses, yeah. Yeah, viruses. There are viruses that infect bacteria. So how do the bacteria defend themselves? They produce, they produce restriction endonucleases that chop up the viral DNA. Now, does that sound like it could be useful? These particular viruses, they're called bacteriophages, and they infect bacteria and can kill them. We have a potential here for treating bacterial infections. When we run out of antibiotics, the trouble is the bacteria do become resistant to these phages. But there is potential for, and it's on the bottom of my list over there, phage therapy. And it is used in some countries. They use phage therapy. They give people injections of bacteriophage viruses to kill the bacteria in their bodies. So the viruses will invade the bacteria and kill them. And because that's the only place the viruses can live, once the bacteria are gone, the viruses go. They don't invade human cells. They're bacteriophages. So they tend to be very specific, I think, to certain infectious bacteria. So, you know, it has got real potential, but there is this problem with the bacteria developing a resistance. And yeah, always the way, isn't it? You know, you have to be one step ahead all the time. So that's where these enzymes come in anyway, that bacteria can produce these enzymes that snip up DNA in very particular ways. And so you can cut out a gene using the right sort of enzyme. Now, the enzymes are named according to which bacteria they've come from. The base sequences are very specific. It's actually in itself just restriction endonucleases would make a presentation, to be honest. But yeah. I'm not going to put that on the list at the moment. A third option, I'm sure there's a fourth one, but I couldn't remember it yesterday either. The third option is that you start not with DNA at all, but you start with messenger RNA. Now, if you can culture cells like, let's say, some pancreatic cells that are producing insulin, as they produce insulin, each cell will only have 2 copies of the insulin gene, won't they? They'll have 1 on each of the chromosomes, so they'll have 2 copies of that. So you could take a cell, you could cut out the insulin gene, whoopee-doo, you've got 2 copies. However, what it will be producing a lot of is messenger RNA. And you can actually track that quite easily. And that's what they do with the insulin. Well, we always give the impression that it's the human gene that's cut out and put into the bacteria, but actually it's not quite like that. What we do is start with the messenger RNA and convert that into a single-stranded piece of DNA, what we'd call complementary DNA. And that is the reverse of what? All right, going from RNA to DNA then, is the reverse of DNA to RNA. DNA to RNA was, in protein synthesis, the first step. Forgotten it already. Transcription. Yeah, transcription. It's the reverse of transcription, isn't it? Yeah. And there is an enzyme called reverse transcriptase that can do that. Now you find that enzyme in certain types of virus. Yeah, like HIV, for example. HIV contains RNA and reverse transcriptase, puts both into a cell. The reverse transcriptase then turns the RNA into DNA, and then that can be put into your genome so it churns out more virus. Oh, it's all such clever stuff, isn't it? You know. But if you start with the messenger RNA, then I'm going to do this as a little oh, I've not left myself enough room to write the enzyme on there, never mind. Single-stranded complementary DNA. Sometimes it is just given the little initial C in front of it, cDNA. It's like with recombinant DNA, you'll sometimes find rDNA. Because, you know, identifying these is quite important. So we have this enzyme, reverse transcriptase. That's not going to fit in. There we go, reverse transcriptase. So has that then made some DNA? Well, it hasn't, no. You know, you've got some DNA stuck to some RNA. That's not DNA. That's not double-stranded. But you can then build the other strand of the DNA just using which enzyme builds that? Yeah, DNA polymerase. Yeah. So then convert to double-stranded DNA using DNA polymerase. All right, so I'm sure somewhere there was a fourth option here, but I can't remember what it was. I may be making it up in my head, but basically at this point we've got perhaps a few copies of the gene. We might have a few hundred, perhaps, if we've used that last method, but we probably want a lot more than this. So the next process is magnifying or amplifying is the word, sorry, amplifying the gene or cloning it, making multiple copies of it. And this is where that process comes in that you had in that set of dominoes, the polymerase chain reaction. All right, PCR. Now, I'm not going to go fully through that today because I want to use it as a sort of pick-up point tomorrow morning. But we've got, trying to think how to describe this. Right, I had just a dash, didn't I, for obtaining the gene. So amplifying, which just means making more, or cloning the gene. You could introduce it into some bacteria and let them just keep copying it, which is fine if you want it to go into bacteria anyway, because you're not going to get it out again. You know, if you're doing the GM insulin thing, then yeah, you'd only need to modify. Well, really, if you took it back to basics, you only need to modify one bacterium, and then yeah, they multiply so efficiently that you'd get loads of copies, wouldn't you? But you might, there are a lot of situations where you need multiple copies of the gene, and this isn't just for genetic modification. It's also for things like DNA analysis. So for things like genetic fingerprinting, for example, where you might go to a crime scene and you might find one hair with a few cells on the bottom end that you could extract some DNA from, but there's just not enough to put through the analytical technique. So you need to multiply it up so that you can actually process it, so that you've got something you could actually see. So we need a mechanism for doing this, and this is where the polymerase chain reaction comes in. Now, I realise I must have seen a demonstration of this not long after it was first available. And basically, it's just a machine that actually does the polymerase chain reaction. Any analysis is a separate process. But the polymerase chain reaction, polymerase refers to DNA polymerase. And it's completely artificial. Right? You put your DNA sample into a machine and it heats it up. That separates the two strands, because we're not talking about a protein and denaturing. If you heat it up, it'll just break the hydrogen bonds between the two strands. You then introduce DNA polymerase. No, you don't do that straight away, sorry. You have to introduce some little fragments of RNA, little single-stranded bits that will stick to the DNA and give you a starting point. We had the same thing in replication. When we did the DNA replication, you had what we called a primer involved. So they put a primer in to start with, that will bind to the DNA. You then put in nucleotides and DNA polymerase, and it will build a matching strand. So once you've put a single molecule of DNA through one cycle of this, how many have you got? Which is simple, 2, yeah? Once you run it for a second time, you've then got 4. And then 8, 16, 32, 64, and so on, until you get into millions very, very quickly. And you've then got material that you can work with. So it's a really, really easy way and it is not complex technology, this bit. So you start with your single one, separate those, you end up with, you build two, then you it is really multiplying up the DNA molecules very, very rapidly. Now I think just to tidy this bit off, I've got an animation for this, and it's one of the things I want to use. So, I'm just thinking you're going to lose a bit of tomorrow morning with this person coming in to talk to you. So I need to get a bit further with you than I did with the other group. I'm just trying to think where my link is for this. I think it's on. Oops. Bear with me. Just trying to think where. I've got a link to it on a lesson plan. That's no use whatsoever, is it? PCR animation, the McGraw-Hill one, and I know how much we love those, but it is actually quite a decent one. There are loads of others as well. Actually, there's probably a better one on your genome. I will have a check at that one. I don't think this will be very long.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
Okay. All right, that's that's one of them. It's and there's nothing really complex about this at all. So, oops, that's not what I meant to do. I want that down there. The other good source is on your genome. Now, on here. Oh, it doesn't run on here, does it? Sorry, got to run it in Chrome. On the Your Genome site, there is also an area that is like sort of simulated practical work where you have to click on something, lift it up, and drop it somewhere else. And there's one of those for the for this, but it's not very spectacular. You just click on a test tube and move it to the thermal cycler, and that's it, really. But on here, sorry, this will just take me a minute to. Oh no, it should pick it up, shouldn't it? Because I haven't. Oh, are you kidding? Doesn't it recognise that yet? Right. It's not the sequencing bit I want, but I can get there through this, can't I? Sequencing is, part of what we want, but I just want to find. It's not going to be there, is it? Somewhere on here? Okay, it's not there. Somewhere in there, I'm sure There's PCR. Okay, methods and technology maybe. PCR, there we go. That's a facts. I wanted to. Oh, I was sure there was a video of it. Maybe not. I've found one somewhere else, but yeah, there is more information on there anyway. So you've got, yeah, it says developed in 1983. Yeah, it wouldn't have been long after that I saw a demonstration of it at the Wellcome Institute, and so you've got the information about it here, but it really isn't particularly complex. So the technique is a fairly straightforward one to understand. Okay, that's a sort of tidy place to stop. What I'm going to go on to next time, so tomorrow morning after we've had this person talking to you, seems to think it'll only take 15 minutes. And then it says, depending on how chatty the students are. So, keep them talking. No, keep them talking and say, it's preparing you well for whatever you're going on to and all that sort of thing. But could do with a bit more practical. Then we will just have a look at how you would transform a cell. In other words, we'll look at the bacterial example, these plasmids. It's not complicated. I can do a little bit more on the restriction enzymes, I think, at that point. And then culturing them and identifying the ones that have been modified. And then we can go on and look at DNA probes, and I'll leave the assignment bit until tomorrow, I think. Hopefully we'll have a few more people in as well by then. Okay, so we will leave it at that.